Hey Misha,
Hello, Farah.
have you had your dinner?
Sorry?
Have you had your dinner?
Not yet. Later. You?
I already have.
Oh, that's great.
Hi Misha, hi Farah.
Hi, Carmen.
Hi, have you guys done with this week's assignment?
We have so many assignments. What we have to do for hematology assignment?
I think we have to find the following topics in hematology. For instance, the introduction, fun facts, terminologies, composition and function of blood, haematopoiesis of process, quality assurance, what else? Quality, crucial and standard operation procedure in hematology. I think we have to find that.
Wow, there's so many of that.
Right, yeah.
Yeah, so we are going to describe and explain in detail for each section for these topics, right?
Introduction of Haematology. Haematology is the study of blood and blood disorder. These include blood and bone marrow cell. There are common hematology tests such as complete blood counting that used to aid in diagnosing anemia, certain cancer of blood, inflammatory disease, and to monitor blood loss and infection. Second hematology test is platelet count, is to diagnose and to monitor certain type of bleeding and clotting disorder. Lastly is bone marrow biopsy. This test involves removing a small sample of the bone marrow inside your bone for testing using a small needle insert into your bone.
These are the 3 fun facts that we learned throughout this assignment 1.
Next, I'll be explaining the basic terminologies in hematology. The word acute means that the disease progresses very quickly, sudden and less severe and also reversible, whereas the word chronic is often slow progressing, more severe and irreversible. The next word is bone marrow, which is the soft spongy tissue inside our bones. It is also where our blood cells are produced. The next word is anticoagulant, which is a drug that prevents blood clotting. Different tube contains different preservative and is used for different function. For example, the green tube is contains sodium heparin and it is used for biochemistry tests. Next word is blood smear or film, which is a glass slide used to observe under microscope to determine blood's morphology and also number. Next, we have the word artery. To make it easier, artery is a muscular vessel that carries oxygen, nutrients, hormones, and other chemicals throughout the body. And vein, on the other hand, is the blood vessel that carry blood flow in oxygen away from the body and back to the heart. What's in between artery and vein is called the capillaries. They are the smallest blood vessels that connect arteries to veins. The next word is called leukocyte, which is also called white blood cell. They are the soldiers of the body and they protect the body from infections. There are 5 types of white blood cell. The first one is basophil. Basophil is in charge of all parasitic infections and allergic reactions. It increases with bone marrow damage. The next white blood cell is called eosinophil. It is a type of white blood cell that mediates allergic reactions and defends body from infection. The third white blood cell is called neutrophil, which is another type of white blood cell that has the highest number in the body. It helps to fight infection as well. And the fourth white blood cell is monocyte. It ingests bacteria and foreign particles. All the 4 white blood cells mentioned just now, they belong to the body's innate immune system. And the last type of white blood cell is lymphocyte, which is the body's only white blood cell that is part of the body's adaptive immune system. Lymphocyte consists of T lymphocyte and B lymphocyte. The T cells regulate the function of other immune cells and directly attack infected cells and tumors, whereas the B cells produce antibodies. Besides white blood cell, there's also red blood cell, which is called erythrocyte. It transports oxygen protein that carries hemoglobin. The next word is granulocyte, which means the small granules within the white blood cell. There's only 3 white blood cells that contain granulocytes, and they are basophil, eosinophil, and neutrophil. Okay, next we have the word plasma. Plasma is the liquid component of blood that transport blood cells throughout the body, along with nutrients, waste products, toxins such as urea, ammonia, creatinine, and they transport antibodies, proteins, and chemical messages throughout the body. Then we also have plasma cell, which is part of white blood cell, particularly lymphocyte, B cells that make large amounts of antibodies. The next word is thrombocyte, which is also called platelet, a small fragment cell that prevents overbleeding and it's involved in blood clotting process. The next word is blood, which is a liquid connective tissue that contains cellular elements. Blood carries oxygen and nutrients to other tissues. They form clots in response to injury and carries defensive cells and antibodies that fight infection. The next word we have lymph node, which is a small bean-shaped structure throughout the body. It's part of the body's immune system that contains white blood cell or the lymphocyte that functions to filters foreign substances through lymphatic fluid, and it helps body fight infections and diseases as well. Okay, next we have the word erythropoietin, which basically is a type of hormone that is secreted by the kidneys to control the production of red blood cell. Haematopoiesis is another word that you hear a lot in hematology class, which basically means the process of blood cell production and maturation. Whereas hematopoietic, also called hematopoietic stem cell, is the immature blood cells, the blood cells that have no identity yet and have not developed into either red blood cell, white blood cell, or platelets. The next word, we have the circulatory system, which consists of the heart and blood vessels for transporting blood throughout the body. So the peripheral blood is the blood circulating through vessel. And in hematology, blood count is very important. Hence, the complete blood count is a test that provides the types and numbers of cells in one's blood. This is also the type of test that can identify diseases like anemia and other infections. So besides complete blood count, we also have white blood cell count, which measures the number of white blood cells in the body. This can help to identify and detect any problem with our body's immune system. The next word is clot, which is also called thrombus, basically a clump of platelets and blood proteins. It is also called the aggregation of blood. The clotting factor is important as it can help to prevent excessive bleeding. And the next word is coagulation, which basically means the process of blood clotting. Next, we have the word hematocrit, which is the calculation or the percentage of whole blood volume that is made up of red blood cells. And next is the word haematology and haematologies. Haematology is the study of blood and blood-related diseases, whereas hematologist is a specialist that specializes in researching, diagnosing, and treating blood disorders. The next word is hemoglobin. Hemoglobin is a protein in red blood cell that carries oxygen to other cells in the body. Next terminology is leukemia. Leukemia is a cancer that is found in blood and bone marrow, and it's caused by the production of abnormal white blood cells. So as you can see in the slide, the normal blood, we have more red blood cells, whereas patients with leukemia, they have abnormal number of white blood cells. And the production of white blood cells are also abnormal. Next, we have the word anemia. Anemia means that someone who does not have enough red blood cell or their red blood cells do not function normally. There are some symptoms of anemia. You can refer to the slide, and also you can see from the picture that a person with normal red blood cells, we have a lot, but on the other hand, an anemic patient, their number of red blood cells are much reduced compared to normal persons. Next, the word myelogenous means that the disease of blood-forming cells in the bone marrow. So the last terminology, we have the word neutropenia. Neutropenia is basically it means that the number of neutrophils are very low. So you can see from the picture, a person with normal neutrophil range and a person with low number of neutrophils. Next, I'm also going to talk about the composition of blood. Basically, our blood is made up of 4 main components. We have our white blood cells, red blood cells, the platelets, and also plasma. So, white blood cells, there are 2 categories, granulocytes and agranulocytes. For granulocytes, it consists of neutrophils, basophils, eosinophils, as mentioned earlier. And for agranulocytes, it consists of monocytes and lymphocytes. As for the plasma, we have amino acids, nutrients, proteins. The proteins include albumins, fibrinogen, and globulins, and also some other plasma, such as nitrogenous waste, electrolytes, and gases. Next, I'll talk a bit more about the breakdown of blood or the blood component. About 8% of our total body weight is actually our blood, and whereas about 92% are made up of other fluids and tissues. As you can see from the picture, the whole blood tube, after going through centrifugation, will give us our plasma, Buffy coat, and also form elements. And you can see that the plasma makes up the most of our blood, which is about 55% of our blood. It is pale yellow in color and also the liquid state. The blood plasma is important because it consists of some nutrients, water, enzymes, salts, and also other proteins and components that are important for our overall health. Okay, next we have the Buffy coat. The Buffy coat is less than 1% of our total blood. It contains leukocytes and platelets, whereas the erythrocytes, red blood cells, they are about 45% of our total blood. As you can see, plasma has the highest percentage of blood. Out of the 55%, 7% is actually protein, such as our albumins, globulins, and fibrinogen, prothrombin, and 91% is actually from the water, and other solutes are only 2%, such as nutrients, waste products, gases. So at the bottom there, we have the form elements, which is our erythrocytes, red blood cells. They, out of the 45%, more than 99% of the form elements are red blood cells. At the bottom of the slide, you can see the pH scale. The normal pH of blood is actually between 3 to 7.4. It is slightly alkaline. Whereas for the optimal temperature of the blood, it's at 37.5 degrees Celsius. Next, I'm also going to talk about the functions of blood. The blood has the following functions such as transportation, regulation, protection, and balance. For transportation, it provides oxygen to the cells. So this means that there is gaseous transportation between oxygen and also carbon dioxide, where the blood absorbs oxygen from the lungs and then transports to different cells of the body, and the carbon dioxide will be removed from the blood to the lungs and then exhaled. The blood also transports waste products to the kidney and liver. The blood will enter the kidney where it is filtered to remove nitrogenous and all toxins products out of our blood plasma. Next, the blood also transports hormones and nutrients. The digested nutrients such as glucose, vitamins, minerals, and including proteins, they are absorbed into the blood through capillaries. And as well as for the hormones, they are secreted by the endocrine glands and transported by the blood to different organs and tissues. The next function of blood is regulation. Blood helps to regulate our body function and also for heat distribution, and thereby maintaining our body's homeostasis. So it helps to maintain our internal body temperature by absorbing or releasing heat. The next function of blood is for the protection of the body and also defense against diseases. The white blood cells, they are known as the soldiers of the body, and they will protect our body from harmful microorganisms, from pathogens and infections. As for plasma protein, these are the protein that contains antibodies that will help our body to fight infections. Last but not least, the function of blood also helps to maintain a balance, to maintain proper water balance in the body, as well as to act as a buffer and help to regulate our body's pH and body temperature. Maintaining the electrolyte balance is important for blood clotting to happen at the site of injury. The platelets along with the fibrin form the clot at the wound site, and this helps to prevent overbleeding.
Haematopoiesis process. Haematopoiesis is the process of creating a wide variety of blood and bone marrow cells, namely erythrocyte, platelet, granulocyte, lymphocyte, and monocytes. Multipotent hematopoietic stem cells are able to divide into increasing specialized cells, a process which repeats and eventually leads to mature white blood cells, red blood cells, or platelet. This series of division in hematopoiesis creates a chart with several branch points, beginning with the multipotent progenitor cells dividing into either a common myeloid progenitor or a common lymphoid progenitor. Common myeloid progenitors eventually go on to create megakaryocyte, erythrocyte, basophils, neutrophils, eosinophil, and monocytes. Common lymphoid progenitors will produce natural killer cells as well as B and T lymphocytes. Multipotent progenitors and any cells the progenitors create lack the capacity to self-renew and must instead always divide into a further specialized cells.
Today I'm going to talk about the quality assurance in hematology. What is quality assurance? Quality assurance is the totality of features that characteristic of a product or service that bear on its ability to satisfy stated or implied needs or reliability of the result in a given condition of performance or expectation. Hematology lab quality assurance, we have three, which is pre-examination, examination, and post-examination. Control planning in lab, we have five: define, measure, analyze, implement improvements, and then control the performance. As you can see, these are some quality controls. SOP should be prepared for every analytic test undertaken and for all significant activities relating to the practice of the laboratory. First is specimen collection. Identify the patient, offer the patient to sit that the patient feels more comfortable, and then explain the process of venipuncture. Prepare the 70% of alcohol, swab, tonic, suitable needle and syringe. Select the best vein, tie the tonic, and palpate any potential way to determine the signs, direction, and depth. Rub the alcohol swab in the circular motion, moving outward from the site. Penetrate at 30-degree angle in one swift, smooth motion to decrease patient's discomfort. Release the tonic as the blood enters the syringe and slowly pull out the piston to get as much blood needed. And then withdraw the needle and clean the puncture site with 70% of alcohol and transfer the blood onto the appropriate tube. Mix the blood with anticoagulant so the blood is not lysis. Next is lab safety. We need to wear lab coats and then wear close-fitting disposable plastic gloves. Never take personal items such as cosmetics, food, drinks, or cigarettes. Never perform any action which may bring your hands into contact with your face, eyes, or mouth. Do not taste or intentionally sniff chemicals. Never consume and store food or beverages.